with most proteins turning over within a day. So there's this constant turnover and new synthesis of proteins. So how is it then, when you have protein turnover, that you actually can maintain the system in this elevated state? So early studies showed that indeed if you blocked protein synthesis using a chemical inhibitor, what you found was that the memory, the synaptic memory here, was no longer persistent, that it came back down, that the increase in synaptic strength came back down to its baseline levels, showing that the synapses actually need access, or they need new proteins for the memory at this level to last. Now, an interesting problem that's posed by the unique morphology of neurons is how they're going to get those proteins. Now, here is a picture of a neuron, and this is the cell body. And the cell body is where the nucleus is, and in the nucleus is the DNA. So the DNA gets made into RNA, and the RNA gets made into protein. And most people, for a long time, thought that all of the proteins that the neuron uses are made in the cell body. So how would it be then, let's say that you're changing a couple of synapses way out here, how would you actually get the proteins that you need to keep those synapses strong? Somehow the proteins would have to be made here and sent out and somehow find their way to just the right synapses. Well, fortunately, there's a solution, and that is that neurons are different from other kinds of cells. They don't just keep their protein synthesis machinery near the nucleus. They actually have protein synthesis machinery way out in the dendrites at the synapses. And this was, again, seen in electron micrographs where people observed polyribosomes. So ribosomes are the protein synthesis machinery, and polyribosomes are clusters of ribosomes that are actively making protein. And in these electron micrographs of synapses, what some anatomists found was, here is a presynaptic terminal, and you can hopefully see the little vesicles here. And here's the dendritic spine, and here is a cluster of polyribosomes right next to the synapse. So this gave rise to the idea that maybe the synapse could make its own proteins right here, and so then you would no longer have this trafficking problem of shipping a protein that was made in the cell body all the way out to the dendrites. This is something that we have worked on extensively in my own group because early on we did experiments with the neurotrophic factor, brain-derived neurotrophic factor that's in that family, the NGF family that Rita Levi-Montalcini showed was important for neurite outgrowth. And we showed a form of plasticity that BDNF could elicit that required protein synthesis in the dendrites. And one of the really fascinating and amazing things about being a scientist now is that a lot of what we do does not need, or conclusions we draw are not always inferential. That is, we don't have to infer things. We can now see things. And a lot of this has to do with the discovery of fluorescent molecules in lower organisms that we can use. And one of these you may have heard of is called green fluorescent protein. It's a protein from jellyfish that we have the gene for, and now we can use that gene and hook green fluorescent protein up to any kind of process that we want to see, and we can actually visualize it. So it's a very rewarding and immediate feedback kind of experiment. So we wanted to see new protein synthesis in neurons, and we wanted to visualize it. And so what we did was to take the gene for green fluorescent protein and express it in neurons. So we put the gene into the neurons, and we made sure that the messenger RNA for green fluorescent protein was sent way out to the processes here. And then what we did was to actually do a little microneurosurgery on a neuron, and we cut this part of the dendrite away from the cell body. So that way we showed that any change in the level of GFP that we could observe out here could not be coming from the cell body because we had disconnected the dendrite from the cell body. So in these experiments, what we showed, this is the dendrite before and after the application of this growth factor, is that that growth factor could stimulate protein synthesis in the dendrites. This is the same neuron here where the fluorescence intensity is encoded by changes in the height and the color of the pixels. So this is the dendrite before and after BDNF. So those experiments showed definitively that dendrites can make their own protein, which of course allows them with endows them with a great deal of independence. Now they no longer necessarily rely on the cell body, and you can see quite easily now how individual synapses can function independently from their neighbors. Now, there are other ways that neurons change. Not only can you change the complement of proteins that are in a synapse, you can also change the shape of the synapse. And by changing the shape of the synapse, you can either increase the contact area that the postsynaptic cell has with the presynaptic cell, or you could form a new connection. So here's just a close-up of the dendrite, and these little pearl-like structures that you see coming off of the dendrite are the dendritic spines. And those are the areas where the presynaptic nerve terminals that we can't see in this image come up and make a close contact. So the synaptic cleft is right here. And these are all the structures that are specialized, much like the folds in the brain, to increase the synaptic contact area of a given cell. And so you can also, this is again using green fluorescent protein, and the reason it's not green is that we code for the intensity, so the more protein that's there, the hotter we make the color. So that's the yellow is the saturated color. And so we can zoom in and actually take movies of how the synapses are changing over time. And here's a little movie that was taken by a graduate student of mine. So this is an individual spine changing its shape over the course of an hour. Let me just go back so that I can show it again. So it's really fascinating. This is the kind of dynamics that's going on all of the time in your brain. Spines are changing shape, and new contacts are being made, and we can actually visualize that now directly. So that's what I wanted to say about the sort of cellular basis of memory. And now I'd like to talk about how we approach studies of memory behaviorally. So when we think about memory, it's such a complicated topic to think about. It's best to try and break it up into components that we can address experimentally. And so if you think about a memory, you can think about the encoding phase. That is, how is the memory initially laid down in the brain? How is it stored and turned into a long-term memory? And then, of course, how can we access it? If we can't pull the memory out, then it's somewhat useless to us. So that's the process of retrieval. And there is a sort of temporal, there's a timeline here where, of course, encoding is the early process, then consolidating the memory, and then pulling it out at variable times. Now, in addition to these different phases of memory, there are different kinds of memory. And studies looking at the contribution of different parts of the brain have helped us understand that there are different ways that the brain interprets the memories that we form. And neuroscientists like to think about memory in two broad categories. There's one category that's called episodic or explicit memory, and this is the memory that most of us think of as humans. This is our memory for facts and events, like remembering the birthday party of your grandfather or a special family celebration, things like that. Implicit memory are more sort of automatic kinds of memory, and those are things like learning how to ride a bicycle, you never forget that, simple Pavlovian conditioning, and also non-associative learning, which is learning to habituate, learning to forget things. So, for example, an episodic memory for you, some snapshots might be something like this when you came in to the Congress Center today. So this might be kind of your